So this presentation is going to be a bit different, talking about a very different kind of cohort study that we have had going in Hawaii since the early 1990s. And it's a collaboration between the University of Hawaii Cancer Center, my institution, and the University of Southern California. But first, I'd like to dedicate this talk to the multi-ethnic cohort participants who perished in the Maui fires. And it was a very sad news, obviously for this community, but for the entire state and many people around the world. So talking about the justification for a multi-ethnic cohort study in Hawaii and Southern California, for us it was really the diversity in cancer rates that we see amongst the different ethnic groups in Hawaii. And you have seen version of these slides. This is for the entire state of Hawaii. And we are really unique by the diversity of our population. And we are also leading the country in terms of the proportion, proportion of admixed individuals. So as young generations grow up, we have more and more admixed individuals, and I think it's what the United States is going to see in the coming decades, and that would be for the better. As I mentioned, we see major differences in cancer rates across ethnic groups here for breast cancer. As you can see, the rates are higher in Native Hawaiians compared to whites, and it's lower in Chinese and Filipinos, but we also have seen an increase in rates for Japanese, which started at relatively low rates, are now as high as whites. Major differences in mortality as well for breast cancer, so higher mortality in Hawaiians, but as you can see for Japanese, actually mortality is lower due to a better survival, even adjusting for stage and treatment information. So major differences that we don't understand and could obviously lead to a better understanding etiology of cancer, but also ways to prevent and possibly treat cancer. So our approach is focusing on cancer disparities, but which is usually defined as an adverse differences in either a stage or treatment response by NCI. But we are going a step further. We are interested in differences across ethnic group, and they can be favorable or unfavorable, and understanding them has led us to discover additional disparities that were not obvious just looking at cancer rates from registries. And obviously the goal is to develop interventions to help control cancer and reach health equity. We use in our work many biomarkers to try to really open a window to the biology and understand better etiology. So the multi-ethnic cohort, as I mentioned, is a collaboration between USC and USCC. The goal, again, understanding ethnic differences in cancer risk and outcomes. Our focus has been, certainly in the first 2 decades, on diet and nutrition, as well as genetics. But we have diversified those goals, looking now at social determinant of health, environmental exposures, etc. Our funding has been almost entirely from the National Cancer Institute. It's a cancer cohort. However, we are also looking at other chronic diseases such as diabetes and now Alzheimer's disease and longevity. So we have actually, when we apply for the cohorts, we wanted to include something like 9 different ethnic groups. We had to go through peer review and reality of funding. So that was reduced to 4, and actually we got additional money from the Department of Defense to be able to add Native Hawaiians. So we have 5 groups in Hawaii: Native Hawaiians, whites, and Japanese Americans. And in Los Angeles, Latinos and African Americans. And we have focused on health disparities for the main cancers, but now that we have over 30 years of follow-up, we're able to look at rarer cancers and, as I mentioned, other chronic diseases. This is a website, and the cohort is very well described at the website, so you may want to check that out. Just to give you an idea of the type of information we collected in the cohort, 215,000 people in total. The way it was put together is to mail questionnaires to names and addresses in Hawaii and Southern California, mostly Los Angeles County. We mailed out 26-page questionnaires, and those who filled out the questionnaire and sent it back entered the cohort, and they have been followed since then. The follow-up, I didn't mention that, is done mostly through linkage to SEER registry. So we have a statewide SEER registry here in Hawaii, and there's been one in California as well. So it's mostly passive linkages, passive follow-up. However, we have sent follow-up questionnaires every 5 years to update a number of risk factor information. So the 26-page baseline questionnaire included information on demography, which was quite detailed. We asked about ethnicity and race of the subject as well of each parent, their place of birth. We have obviously smoking history. We have medical history, reproductive history, physical activity, family history, and we have a food frequency questionnaire that was developed specifically for our population, and it was like 22 pages, so there was more than 350 food items, and that was validated against 24 recalls in each of the ethnic groups. Here is what the food questionnaire looked like. We ask about frequency of consumption and serving size, and we help the participants by providing some pictures of different serving size for groups of food items. And those, again, were developed over something like 5 to 7 years by collecting food records and from actually 9 ethnic groups in Hawaii and Los Angeles. As I mentioned, we mailed out follow-up questionnaires every 5 years to update risk factors, which are change over time. But as the cohort aged, we added different questions about healthy aging, social and social isolation, things like physical functioning, frailty. In the last questionnaire, we asked about discrimination and medical marijuana, which is legal in both states. In terms of numbers, we had a total of 215,000, about 57,000 Japanese Americans, 49,000 whites, 47,000 Latinos, 35,000 African Americans, and about 14,000 Native Hawaiians. So a very large cohort, but until a few years ago, we have not been able to really look specifically at maybe specific ethnic groups, but also subgroups among those ethnic groups. Now we are able to do that because of the long follow-up, as a large number of cases we have. To give you an example, now we are looking at Latinos by place of birth, by generation. We are also looking at Okinawans. Amongst the Japanese Americans, we have about 7,000 descendants from Okinawa. And we find some very interesting differences, actually, by place of birth or by subgroups of those groups. So we lost about 58% of the cohort. We have 90,000 survivors at this point. When we started, they were between 45 and 75, and now the median age is 84. So so as I mentioned, we shifted some endpoints. We are now able to look at healthy aging and Alzheimer, and I think we have something like 2,500 centenarians. So we're starting to look at longevity as well. Where did our participants live in Hawaii? So half the cohorts in Hawaii, half at Los Angeles. We cover all the main islands, and we had 8,000 on Maui, including 14,000 in Lahaina and Kula, the location of the wildfires. In terms of samples, biospecimens that we collected, we got blood from a subset of 70,000, so a large repository, urine, about the same number, mouthwash from people who refuse the blood samples. We give them the option of getting a mouthwash or saliva. We also collected viable lymphocytes on the subset in Hawaii. We got a supplement to our grants to do that. And actually, they are being used by my colleague, Dr. Monak here. So it's really nice to see that. We have collected stool samples on 8,000. And we have done some work on gut microbiome and diabetes, obesity, and different endpoints. So all that is stored in Hawaii and USC, about half and half. In terms of research impact, just looking at number of publications, there are about 1,100 publications so far from the start of the cohort. So those are papers using the cohort from what we call MEG collaborators, which are really USC, Hawaii, and UCSF. We have a group at UCSF that use the cohort. And I have to say that many of those were actually trained. Many of those people were actually trained on the MEG. They did their graduate studies or postdocs using the MEG data, and they have moved to other institutions. They have their own grants now, but they often are still using the MEG data, which is really nice to see. It's really a multi-institutional project, and it's functioning very well for reasons that I think derive from, I think what was mentioned this morning, the investigators who started the cohort, who really thought that it was something that has to be, if not community-based, certainly a community-oriented cohort, and really trying to bring up people from those communities to work and do research with the data. In terms of incidence of cancer, we see the same differences as in the state registries. So high rates of lung cancer in African Americans and Native Hawaiians, high rates of breast cancer in Native Hawaiians, high rates of prostate cancer in African Americans, and high rates of colorectal cancer in African Americans and Japanese Americans. So there's not like one site which is high, one ethnic group which has high rates for all cancer sites, or one that has low rates for all cancer sites, so, which makes the data even more valuable. We were interested in smoking because we saw some early on some differences in lung cancer risk, even after adjusting for lifetime use of cigarettes. So it's adjusting for pack years, duration, age started. So all risk factors for lung cancer, we still see some major differences. 50% higher risk in African Americans and Native Hawaiians compared to whites, and 50% lower risk in Latinos and Japanese Americans. So obviously that was very puzzling, and we did quite a lot of work trying to understand reasons for those differences. We think we explained some, but certainly not all. We are unable to explain the high risk of lung cancer in Native Hawaiians. It's not just due to smoking, there is something else going on. We're still working on that after 20 years, but it's the research life. However, I think we understand pretty well the low risk of Japanese. We looked at nicotine metabolism, which is mainly performed by an enzyme CYP2A6 in the liver, which show differences in activity across individuals, and people with high activity, metabolize faster than nicotine. They need to replenish their levels of nicotine in the blood. So they have a tendency to not only smoke more cigarettes, but also smoke them more intensely, so get more nicotine from each cigarette and more carcinogens at the same time. So we hypothesized that they were at increased risk of lung cancer. What we did was to look at genetic variants that are associated with CYP2A6 activity. And in a large consortium of lung cancer, we were able to show that those variants that seems to determine your level of CYP2A6 activity also are associated with lung cancer. So now we are looking at whether this might be helpful in identifying high-risk individuals, high-risk smokers, for screening to improve the performance of screening for lung cancer. In terms of breast cancer, when we looked at the men known risk factors for breast cancer and adjusted the rates compared to whites, you can see that the differences are reduced, except for Native Hawaiians. Again, we don't understand why they have higher risk of breast cancer, and certainly not known risk factors. And we have been doing some work. We have focused on obesity, and as you know, obesity is usually measured by the body mass index, both in research, in epidemiologic research, as well as in the clinic. And this is clearly not a good measure of obesity, since it it not only measures fat, but also lean tissue, and it regroups different types of phenotypes, adiposity phenotypes. So we went back and we studied 1,800 MEG participants, as many in each ethnic groups, covering a range of BMI, and we have them undergone abdominal MRI and total DEXA. So we have very good measure of total adiposity, distribution of fat, and visceral fat. So the fat that is intra-abdominal, around the organs, and that is known to be more metabolically active. It's strongly associated with diabetes, heart disease, and we thought cancer, or at least some cancers. So we studied about 1,600 people, and we found some differences across ethnic groups, with Japanese Americans and Latinos having more VAT, and Native Hawaiian as well, more VAT, and African Americans, which is on the left, having lower levels of visceral fat. So for the same level of total adiposity, so we're really looking at fat distribution. So, when you accumulate weight, it seems that Asians and Native Hawaiians have a tendency to accumulate their fat, deposit their fat more intra-abdominally. So fat, again, which is more metabolically active. So what we did was to develop a score using weight, height, as well as some biomarkers, such as total cholesterol, triglycerides, leptin, adiponectin, inflammation markers. And we were able to predict visceral fat among the entire cohort and look at association with cancer risk. And we found an association with breast cancer, adjusting for BMI. So it's added risk factors for breast cancer. If you deposit your fat intra-abdominally, you increase your risk beyond expected from your BMI. And this association was even stronger in Japanese. So we think it might explain, at least in part, the higher risk of breast cancer in Native Hawaiians and this increase in what we saw over time in Japanese Americans. Because Japanese American, unless Japanese in Japan, have gained weight over time, and we think have deposited this body fat intra-abdominally. As I mentioned, now we are looking at environmental and social determinant of health. We have done that mostly based on address, so using geospatial data. We have a very detailed address history during a follow-up by sending newsletters and requesting a change of addresses. So we have kept all addresses of participants, so we can build really a history of residency and linked exposures in terms of various factors, such as in terms of such as neighborhood socioeconomic status, such as policy at the level of the county for tobacco, for example, various environmental factors that have been measured, especially in California, at the neighborhood or zip code levels, as well as things like air pollution. So various measures that we were unable to ask through questionnaires. In terms of air pollution, we actually found an association in Los Angeles with where people live. If they live close to major highways, their risk was increased. And with EPA data, we have now shown that they have an increased risk associated with particulate matter 2.5. So again, possibility of doing some work that we were unable to do previously. So I mentioned three areas that we talked about, that we worked on. Diet and cancer, I didn't say much about it, but it's something that we have worked on the first 20 years of the cohort. Those findings have been used for dietary recommendation in the US and internationally. We have a very active program in genetics looking at common variation, but we are starting to look at rarer variations now. It seems to be particularly important in prostate cancer. And as I mentioned, geospatial multi-level variables. But we have a good number of other initiatives, such as on liver cancer, infectious agents, multiomics, and as I mentioned, aging and Alzheimer's now. Our data are accessible, available through request. You can go to our website. There is a process to do that. You can apply, and we share our data widely all over the world. And we have done that with about 100 institutions all over the world. And I will stop here and thanking the participants and my collaborators. Thank you very much.